All right, guys, can you hear me in the back? All right, so if any problems here, and just raise your hand so I can make it louder here. All right, let's start. So my name is Yuri Polikanov, as you probably already know. So I will be teaching you second and fourth modules in this biochemistry course. So the first and the third is taught by Professor Jeffrey. All right, before we go into the topic of today's lecture, so let me just make several general announcements. First of all, so this course consists, as you know, of 4 modules, right? So each of the modules would be ended with the exam. So the final exam, which is the module 4, right? So it's not like exam for the entire material. It's not cumulative. So each exam is for just that module, so your life is easier than, let's say, my life when I was sort of at your stage. So we actually had to pass biochemistry exam, not just for 1 semester, but actually for 3 semesters in just one big exam. All right. So there's no curve in this class, right? So there are predetermined cutoffs that you can find in this, like on the blackboard, in a file called course info. You can see it, right? So they're like right here. So there are cutoffs. I mean, so if you're above the cutoff, I mean, you can almost sort of be sure that you will get a grade which is specified. The only way to get extra credit is through, actually it's also listed there, through doing homework. So you did it with Professor Jeffrey, right? So I'll post my homework on the blackboard like shortly, so you will have it. So you will get actually 20, I mean, you can get maximum up to 20 extra points, so which is like extra 5% on your percentage. So again, if you know material super well, I mean, and you're confident, you don't need to bother with this homework because you will get 90% anyhow. So basically, theoretically, you can get 105%, right? So, I mean, this is the way how we basically issue extra credits. So again, I want to mention this in the beginning, right? So because at the end, there would be a bazillion of emails saying, oh, what I can do to get extra credit. This is what you can do. Please do the homework. So this course is lecture captured, basically. So, right, so you can watch videos of the course. Okay, so the lecture capture is not intended as a replacement for physical attendance to the lectures, right? So it's mainly intended for help with reviewing the material. So I'm actually very pleased to see so many of you in the class. So I mean, I really hope, I mean, that this trend will continue and maybe even more of you will show up. I mean, my personal experience shows, I mean, that those who were physically in the class here, right, so, and interacted with me, you can ask me questions after the lecture as much as you want, so I'm here for you. So those students usually perform better on the exams, just because, like, we can address any questions that you may have that are particularly sort of important for the comprehension of the course. All right, so what do I have for you on the blackboard? So you will find the study guide. So basically, study guide is just a set of questions that you can see. If you know the answers to all these questions, you should get A, no problem. So you should get above 90 with absolutely no problem. So if you know how to answer these questions. So, and again, the answers to them will be provided during the class. So one thing which I want to tell you right away, so the exams, on the exam, I'm not going to be asking you anything that we haven't talked in the class. Even if that, I mean, so for me, what's most important is your conceptual understanding of the material, right? Rather than knowing of small facts, I mean, about something which is not that important. Okay, so something which was in the textbook but was not in the class, I mean, that's not going to be on the exam. So also you will find the self-practice file. So what is listed here is just, I mean, the chapters that we go with the like with their titles. And here, suggested self-practice problems, these are just the numbers of problems at the end of each chapter, right, that you may wish. I mean, it's not, again, this is not a homework. This is just for yourself. So that you may actually want to go through these questions and see if you can solve them. To make your life easier, here, you probably saw that already. I don't know if you've already been at the blackboard, like in the middle. That it says like chapter 6, Leninger problems and answers. Okay, so these are the files that I can have access as an instructor in the course from Leninger publisher, basically. So these ones will have the, like this, detailed answers to the questions after the chapters, right? So because, I mean, in the textbook itself, at the end, you will see only, let's say, there is an enzyme kinetics problem, and the answer should be 5, and you see number 5, but how you get there, so you have no idea. So that is what should help, actually, like with all these layouts, I mean, and so this is just detailed answers to the questions after the chapters. So take advantage of that. And, yes? Yeah, exactly. Yeah, it's these are just files, I mean, that you can see on the blackboard. Yeah, so these are the files, I mean, in the middle here. So the first set of files, these are just PDFs of the lectures, and the last set of files, these are PowerPoints. So the reason I provide two is because I like using sometimes this special fonts, I mean, which if you open PowerPoint, many laptops will not reflect them properly, so that why it's easier to use just a PDF sometimes. That's it. So lectures, like in the PDF and PowerPoint formats, also there's problems to help you prepare for the exam. And also, just in case, I have their scan of like just, and then students a couple semesters ago, I mean, just asked me to help them with some problems, and I just written it on a piece of paper. And this is problem 6.8, that's problem 8 after the chapter 6. So that may actually help you to see what is expected. All right. So I don't know, do you have any general questions before we move on to the lecture? No? Okay, let's move on to the, okay, so today we're going to actually talk about the enzymes. So, so with Professor Jeffrey, you studied proteins in detail, so you know that the proteins are polymers of the amino acids, and so this is like the inseparable entity in every living organism. Essentially, the life itself is largely just all about proteins. And today we're actually going to talk about a special type of proteins called enzymes. So what are the enzymes? Okay, so this is chapter 6, and yeah, before we go, actually, I always like put a slide in the beginning of my lectures, which is just a citation by one of the greatest teachers of all times and nations, Albert Einstein, who said that if you cannot explain it simply, you don't know it yourself. So that if you don't understand what I'm saying, right, so raise your hand, I mean, so interrupt, I mean, ask questions, I mean, that's how you can, you should not be ignorant of that. So there are no stupid questions. There could be stupid answers, but questions, no. So please feel free to ask any questions if something doesn't make sense to you. All right, so what are the enzymes? So this is just a super simplified scheme of all biochemical pathways that we have in any living cell of any living organism. Clearly, I said super simplified, I mean, but it looks super complicated, right? And this is actually just the tip of the iceberg of all the reactions that happen in a cell. So each of these reactions is catalyzed by the enzymes. So the enzymes are kind of the biological catalysts of the reactions, I mean, that take place in our body, in any cell of any organism. So as a simple analogy, we can think about enzymes as tools. For example, I mean, there is a screw here in this desk, right? So how long would it take me to remove it with my fingers? Well, I don't know exactly, maybe forever, maybe a million years, but I mean, it's like I cannot even like sort of grab it. So then if I will have a screwdriver, it's an analogy of inorganic catalyst, right? If I have a screwdriver, I can do it faster. But if I will have like an electric screwdriver, I will do it blazingly fast. So the electric drill or electric screwdriver is actually that enzyme or the biological catalyst which makes the reaction of removal of that screw, which without any help will take forever. I mean, so with the help of these enzymes, the reaction will be blazingly fast. So basically, enzymes, I mean, are just the tools for execution of the chemical reactions in the living organisms. So what are enzymes? So we already partially answered that question, so that most of the enzymes are proteins. So, and they're catalysts, and they increase the reaction rates of the reactions in the cells. So one of the important features of any catalyst, as you probably remember from the inorganic chemistry, is the fact that the catalyst is not consumed during the reaction it catalyzes, right? So that essentially you can have one molecule and it can crunch a bucket of substrate. So that the enzymes, the same as any catalyst, are not used up in the reaction that they catalyze. And of course, I mean, they facilitate or make possible the processes which otherwise will not be happening. So we say that enzymes are proteins. However, biological systems know other types of catalysts, which will not be the focus of our lectures, but I mean, it's just something that you may wish to know. So that catalytic RNA molecules, which are also called ribozymes, and so these are the molecules that can catalyze chemical reactions. So the important feature of the enzymes is that the catalytic power or the catalytic ability would be lost if the protein is denatured. So because enzymes are proteins, right? So if you denature the protein due to the high temperature or ionic strength or something else, so if the tertiary structure of the protein is distorted, so the enzyme loses its catalytic activity. Here, just, I mean, again, not for memorization, just for reference, showing you like the folds of, I mean, how many fold the reaction is being accelerated in the presence of the particular enzymes. So you can see that it actually, we're talking about not just 10 times or 100 times, so we're talking about hundreds, thousands to millions and billions of times acceleration of the rates of the reactions. So these reactions will not happen in the biologically meaningful time frame, so if not for the enzymes. What are the, you may actually ask why biological systems or cells do not use something like inorganic catalysts. For a number of reasons. First of all, most of the inorganic catalysts that are used in industry, for example, right? So they catalyze their reactions, I mean, in the very harsh conditions, like super low pH, like super acidic pH, or very, very high temperatures, like thousands of degrees Celsius. So while when we're talking about cells, I mean living cells, we're talking about very mild conditions. We're talking about pH 7, which is close to the neutral, so it's a neutral pH of water. Or the environment, let's say human body is 37 degrees Celsius, close to 100 Fahrenheit. So these are much, much milder conditions compared to the conditions in which the inorganic catalyst can accelerate the reaction rates. Another thing is that the degree to which the enzymes accelerate the reactions is much greater than the inorganic catalyst. So as I showed you in the previous slide, so it's millions, if not billions of times acceleration rate. So while inorganic catalysts accelerated to much smaller extents. Very important feature, which is absent from inorganic catalysts, is that ability to be regulated. So we want the enzymes to do their job when we want it, right? So like on that electric screwdriver. So when we press a button, it works. When we release a button, it doesn't work. So the same is with the enzymes. So cell need to be able to turn them on when needed and turn off when not needed. So and here just the example. So for example, there is one, it doesn't even matter what is that molecule. So, and inorganically, I mean, this molecule can actually decompose to several different products. However, if we're talking about enzymatic reaction, catalyzed, for example, by the enzyme called chorismate mutase, so there is only one single path is possible or available for decomposition. So in other words, I mean, this is a specificity, so that the enzyme is very specific to the reaction that it catalyzes. So it's not catalyzes just all range of the reactions, it's only the particular one. So it makes a particular reaction to go forward. So enzymes are super selective for the substrates. Well, of course, I mean, so this statement is conditional, I mean, because some enzymes are less specific, some more specific. But here's just an example for you. So that in two substrates which visually looks very similar, like L-phenylalanine or D-phenylalanine, for example, when we will talk about the enzyme like phenylalanine hydroxylase, right? So one of them. So the chemical formula, it's exactly the same. Even the structural formula is the same. It's only the chirality at one particular atom here, at the C alpha carbon of this amino acid, so which makes a difference. So it's only the L-phenylalanine which binds to the enzyme, right, and the reaction goes, while the diphenylalanine cannot bind. So the adrenaline, which is chemically somewhat similar or resembles in its chemical structure the L-phenylalanine, the native substrate of this enzyme, can bind to this enzyme, but the reaction does not go forward. So this, that's why, I mean, this molecule would be called an inhibitor, or the competitive inhibitor, because it binds to the same active site but does not exert any reaction. Okay, so let's, before moving forward, let's actually talk about some very important terms that we will be operating throughout today's lecture and the next lecture, as well as through probably the rest of this course. So enzymes act by binding substrates into the active site. So what is the active site? So it's a pocket on the enzyme where the reaction goes. So it's some sort of a cavity, so where a cavity which has some sort of affinity to the molecules, to the substrates of the reaction, and the substrate is the molecule that is acted upon, right? So this is something that needs to be converted. So in the reaction that is catalyzed by the enzyme. So the product, I mean, it's more or less clear. So product is the altered form of the substrate that is generated during the course of the reaction. So, so substrate binds in the active site, is being converted by the enzyme, and the product is being released. So in the process of conversion from the substrate to product, there is so-called transition state. So this is a very transient, high energy configuration that can decay substrate to product, or, I mean, either way. So the general enzymatic reaction can be written like this. E stands for enzyme, plus S, substrate. I mean, first we have the complex between them, right? So, and then within that complex, substrate changes to product, and then so that product is being released from the enzyme. Again, the key concept here is the active site, substrate, product. So that's what I would like you to remember. So the enzymes quite often do not work by themselves. So they need something to carry their reactions. So, for example, inorganic ions. So we'll see for many reactions catalyzed by the enzymes which metabolize nucleic acids, I mean, like DNA, RNA. So those need magnesium ions. So many enzymes actually do care about the inorganic ions which are around them. Some of them bind them in the active site. So, and these ions sometimes are important for the redox reactions that are catalyzed by the particular enzymes. Another level of complexity of the cofactors of the enzymes would be called coenzymes. So these are organic molecules. I mean, relatively small and often derived from vitamins. So that, usually serve as a temporary sort of docking stations for different chemical groups that are being moved by the enzyme. And the highest level of complexity would be a prosthetic group. So a prosthetic group, it's the coenzyme. It's a subtype of coenzymes, right? So which is permanently and sometimes even covalently bound or attached to the enzyme. As far as I remember, in one of the last lectures with Professor Jeffrey, you were talking about globins, like hemoglobin, myoglobin, right? The proteins that carry the oxygen in the blood. Although it's literally incorrect, I mean, to call, it's not literate to call those proteins enzymes, right? But actually, if you abstract a little bit, you can. So you can, so they still have substrate, like oxygen molecule, right? So it binds to the active site. It releases it some way. It doesn't convert it though, but I mean, it binds it and releases. So that heme, right, you can consider it as a prosthetic group because it's permanently bound to the hemoglobin molecule itself. So as a result, so this would be an example for you of something that is very tightly bound to the enzyme molecule. So. And here just some nomenclature. So the enzyme without a cofactor would be called apoenzyme. So, and everything together would be called holoenzyme. So holo means like the entire, like fully functional molecule of enzyme. So here's just a list of the coenzymes. I mean that you can again. I mean this does not need to be memorized. I would like you to memorize only like the concepts here. So what the coenzymes is and what's the difference between just coenzymes and prosthetic groups. So that the prosthetic group is something that binds very tightly or cannot even be removed from the enzyme. So that common coenzymes, I mean, as I mentioned, so they usually derive from the vitamins that we, and the vitamins are organic molecules which we need to sustain life, but we cannot make ourselves. So that's why we need to receive them, those molecules, with food. So you can have it from fruits and vegetables, or you can have them from the dietary supplements. It doesn't matter. So, but all these vitamins eventually end up in different enzymes, which are crucial for the biochemical reactions that take place in our cells. Here is just an example of cofactor-dependent reaction. So one of the most well-studied enzymes, hexokinase. In the next module with Professor Jeffrey, when you will be studying glycolysis, you will see that this enzyme catalyzes the first reaction in the glycolysis pathway, so which is a way to extract the energy from food. So the way this enzyme works is it basically it transfers the phosphate group from the ATP molecule, which here serves as a cofactor, onto the glucose. So, and we'll talk a little bit in more detail about how that happens. But as an example of the cofactor here would be an ATP molecule that delivers the phosphate group for the transfer to the glucose. You saw how many reactions are there in the cells, right? So like tens and tens of thousands of reactions, right? Obviously, each of the reactions has its own enzyme. So, and it would be a big mess if there would be no system for naming those enzymes or nomenclature for them. So, and there is a standard nomenclature which is more than 50 years old, which basically postulates that there are 6 main classes of the enzymes. And these classes basically, so this class, I mean, segregation of the enzymes into these classes is based on the types of the reactions that they catalyze. For example, if it's a transfer of electrons, so then it would be oxidoreductases. So these enzymes catalyze redox reactions. So if they just transfer groups, I mean, so they would be transferases. So if they hydrolyze, hydrolases. So, and there would be 2 interesting classes, lyases and ligases, which kind of do the opposite. One class creates CC, CO, CN bonds, and the other one actually destroys them. So, and there would be also isomerases. Interestingly, the enzyme which, according to this nomenclature, has number 1.1.1.1 is alcohol dehydrogenase. So that's an enzyme which is responsible for the alcohol detoxification. I actually do not know this for sure, but I was always wondering, more like as a joke, that this big IUPAC Congress, where in 1957 they decided upon this classification, probably everyone who gathered there, like thousands of scientists from all over the world, so they probably get drunk the first night, and then next morning nobody showed up to make decisions. So, and they decided to, again, this is my guess and more like a joke, so, and decided to commemorate that as a sort of, because what would be the reason to call this enzyme 1.1.1, like enzyme number 1? Indeed, that's the one. Our favorite hexokinase is, for example, 2.711. So the enzymes usually have several different names. So any enzyme can be called by a trivial name, like hexokinase, right? So it's just somebody who discovered it first time just called it that way. So, but this name does not reflect really what the enzyme is doing. However, any enzyme can be also given a systematic name, something like ATP glucose phosphotransferase. Maybe even more detailed, ATP glucose 6-phosphotransferase, to emphasize that it not just only transfers phosphate to glucose, but to the particular position in the glucose, position 6. So again, so of course, I mean, these systematic names can be very long, given that you need to pronounce the name of the substrate, the cofactor. So many enzymes are referred by their trivial names quite often, but you just need to keep in mind that they can be called either way. And that's especially important for actually many of you who will decide to become doctors or dentists or pharmacists, because when you will be sort of ordering the lab tests, the blood tests for patients, I mean, so you will get results of many tests for activity of different enzymes, and you will get them for this enzyme and that enzyme, so you need to know what actually is going on. Okay. So let's move on to the mechanism of how the enzymes catalyze the reactions. So when that was going on, first let's briefly talk about the energetics. So we can write down, I mean, basically the reaction of conversion from substrate to product in this form, like substrate reversibly product, right? So this reaction would be characterized by the equilibrium constant, so which is just a basically ratio of the molar concentration of product to the substrate in equilibrium, or at equilibrium. So this little hyphen here means biochemical. So in inorganic chemistry, you probably we still had the same K, right? But without this little thing here at the top. Over there, the meaning of that was basically that it's at 1 molar concentration, something at 25 degrees, and so on and so on. So biological means that the pH is 7, so which is not 1 molar. So again, pH is 7. It's the molar concentration of protons would be 10 to the minus 7 molar of the protons, right? So the concentration of water at this pH, of course, I mean, it doesn't matter. At any pH, is pure water would be 55.5 molar. So, and the temperature. So it's like the 37 degrees rather than 25 degrees Celsius room temperature. So that's what this means. So here we're talking about the biological constants. So the free energy change, or the Gibbs energy, right? So also has this little symbol here. This free energy change can be written the same way as in organic chemistry as minus RT, natural logarithm of this equilibrium constant. As we know from the thermodynamics, so if this free energy change is negative, then the reaction will proceed spontaneously. So that is without energy input. So basically it's already, so the substrate has high energy than the product. So the reaction goes while going to product, right? So the energy drops. I mean, so no need to it's like going downhill. So I would like to draw your attention to the word spontaneously. Spontaneously means that it goes by itself, but it doesn't mean that it goes fast enough. So because how fast it actually goes, I mean, this is the thing which I'm actually showing in a very I would say nerdy way on the next slide. So basically, the speed at which the reaction goes depends on how high is that activation energy, which we're getting to. Okay, for one substrate reaction, so the speed of the reaction would be proportional to the concentration and to the first order rate constant. So let's just keep the two substrate example. So basically, the reaction speed will depend on either of these two parameters. So let's say the substrate concentration is constant. Then if we look at what the K is, or the rate constant, so the rate constant actually is expressed using this equation. So and over there, we see this minus delta G with this weird sign. Okay, so this delta G, this is energy of activation. In other words, how high is the barrier in order to get from substrate to product. You can see that the higher the delta G is, again, because this is the exponential function, and this one has a negative sign, so the higher this delta G, right, the lower or the smaller would be the value of K. As a result, I mean, so the higher the activation energy, the slower the reaction will go. The lower the activation energy, the faster the reaction goes. Jumping a little bit ahead, I can tell you that that's exactly what the enzymes are doing. They lower the activation energy so that they accelerate the rate of the reaction by lowering the this delta G. As a result, I mean, the reactions start to go much faster. Okay, so let's get used to this kind of diagrams. So here we just, we call reaction, it's called reaction coordinate on the x-axis. So you can treat it as time, for example. So we start with and on the y-axis we have energy, which is here, just for the simplicity, it's arbitrary values. So we start with the ground state and the substrate, right? So in order to get the product, it has to go through it needs to climb that hill in order to actually then get into product. Simple example. Imagine like we just basically separated you randomly in two pools, like on one side of the room, on the other. So, and I raised the wall here, right? And the task is to get from one side to another, right? So obviously you need to climb over the wall. It's exactly as what is shown here. You need to physically climb up that wall in order to jump onto the other one. So those who are physically trained well will be able to do it, but probably not everyone, and I will definitely stay with those who will not jump around. So what would be an enzyme analogy here? And I think, it's perfectly shown here, would be a ladder or a door in that wall, right? So that it would be much easier to get from one side to the other, because the activation energy would become much lower. So let's see again what the activation energy. So activation energy, it just is difference between the, let's say, the state at which the substrates were, right, and the energy of the transition state. So the transition state, this is a very transient state in which it's not already a substrate, but not yet product. So it's sort of, we will see that example in a couple slides. When you break in the pencil, it's like the pencil which is not yet broken, but it's already bent. So, and this is what the activation energy is. So once the reaction reached that transition state, it actually has equal, probability to either go back to substrate or go back to product. But the chances of getting to product are higher because, then it will drop from here to here, so the difference in the energies would be higher. So, because everything in the universe likes to be in the minimum of energy, tries to get there. Okay, so what the enzymes are doing. So by binding to the by binding to the substrate, so it already lowers, the energy. So it basically, the main transition state, so this transition state of the reaction, now occurs not in solution, but on the enzyme. And while on enzyme, so it's already at a much lower, we'll answer the question as to why the energy is lower in the next few slides. But for now, we just need to take it for granted. So that when enzyme is there, so the transition state has much lower energy. So then in the same transition state in the solution. So as a result, so the lower the activation energy, the faster the reaction. So that's how the reaction will go. And also you see that the energies of substrate and products are the same, regardless of whether this reaction is going free in solution or it's catalyzed by the enzyme. So the very important thing which I would like you to remember is that the enzymes do not affect equilibrium. So in other words, so that the enzymes increase only the rates of the reactions, right? But do not decrease, by decreasing the activation energy, right? But do not change the equilibrium. In other words, the concentration of substrate and products in equilibrium will not change. In other words, so in that example with the wall and the ladder or the door, so by providing you a door or a ladder, so you would be just going back and forth much faster, right? So, but the amount on one side and amount on the other will stay the same. So, so the equilibrium, again, the equilibrium is not changed. What has changed is just the activation energy. And an important sort of consequence of that is that the enzymes actually increase the reaction, both the reaction rates of the forward and the reverse reactions. So the same enzyme, actually in my own lab, actually we quite often use the enzyme which is made by Mother Nature to synthesize RNA. So we use it to actually destroy RNA. So all what you need to do is, according to the equilibrium, is just give it too much of the product, right? And then it will shift the reaction backward. So the same enzyme will catalyze either the forward or the reverse reaction. It only depends on the concentrations of the substrate and the product. If there is too much product, actually it can go backwards. So this is what I would like you to remember, so that the enzymes do not do actual magic. So they just make the reaction go faster without any sort of specificity or exclusiveness towards one reaction but not the other. Okay. So the slow reactions are characterized by high activation energy, as we already know. And this activation energy needs to be surmounted during the reaction. So how does the enzyme help here? So that was actually proposed by Linus Pauling in 1946, who basically postulated that optimal interaction between enzyme and substrate occurs only in the transition state. So basically his idea was that when the substrate binds to the enzyme, it already, in order to form the bonds with the enzyme, like hydrogen bonds, electrostatic bonds, hydrophobic bonds, hydrophobic interactions and van der Waals interactions, right? So during formation of all those types of interaction, the energy is being released. And because, there are many of them is occurring, right? So that's what actually lowers that activation energy. Another very important, actually, aspect from what Linus Pauling proposed is that the enzymes have the highest affinity to the transition state of the reaction, and neither to the substrate nor to the product, but transition state. That can be best illustrated by this imaginary stick case, which you will find in the textbook. I find it very illustrative too. So very simple reaction. Imagine you have a pencil, you're trying to break it, right? So the transition state in here would be that sort of pencil which is not yet broken but already bent severely, right? Of course, it's reversible. If I remove the other hand, it will bounce back. Okay, so if the enzyme would have highest affinity to the substrate, for example, what will happen? It will be even harder to break it, right? Because the enzyme would actually stabilize, and that's what is shown here by this energy going down, right? So then the activation energy would actually become even larger, right? So if for the free reaction it goes from substrate to the transition state, it's only this high, right? But if the enzyme would be binding tightly to the substrate, then it would be, this activation energy would become even higher. It would be even harder to make this reaction going. So, however, if we imagine that this enzyme is actually, it have some affinity to of course, to the original stick, to the pencil, but the highest affinity is to that bent conformation. So it will promote it. So it will make it occur faster. So because, the active site of the enzyme, it's really congruent. It's really has high affinity to that half-bent, pencil or stick or whatever, or substrate. So that's what's called the transition state. And yeah, this is just the same, the main source of this energy, where does this energy comes from in order to lower the activation energy, comes from the binding. So the binding energy helps offset the transition cost. So the transition cost is just the ability to climb that hill, right? So in the case of our example with the wall, so the ladder will provide that energy. So you will be interacting with the ladder rather than with the wall. So, and that will make it much faster, the reaction. So multiple weak binding interaction occur in the enzyme substrate, enzyme product, and mainly in the transition state. So that's what I would like you to, to make notes about. So that this multiple, but many weak interactions, altogether make the decrease of this energy. This energy is being released upon binding of the substrate to the active site of the enzyme. So in this binding interaction, they produce negative free energy change. So that the energy basically of the transition state becomes lower. As a result, it's much easier to surmount that energetic cost. So the height of this hill is called the energy cost to get the reaction going. So the easier to jump over, the faster the reaction will go. Okay. Now I would like to talk about the entropic contribution. So when the molecules bind to the enzymes, it's not only there is not only the enthalpic, which is just the enthalpic component there, which is heat, right? So the let's say the small molecule bound to the active site of the enzyme, the number of hydrogen bonds formed. For each hydrogen bond formed, there would be 3.5 kilocalories per mole release. So that's heat, basically. That's what we call enthalpic component. So what about entropy? So you know what the entropy is, right? So which entropy would be higher, which one would be lower? During the recess or during the lecture right now? During the recess, the entropy would be higher, of course. So the entropy, there are a bazillion of definitions, but one of them could be, it's a number of states in which the system can exist at a given moment. Right now, everybody sits on the seat, right? So, and this is like entropy equals to 1, right? And then actually when the bell rings and the recess starts, I mean, so students start to, like, I don't know, run on the ceiling, so that would be entropy skyrocketing. So in any case, we need to have that, we need to be on the same page here, so what the entropy is. So besides just actually having this energetic component, the enzyme also have the entropic component. So when the two molecules are basically free in solution, in order to react, in order for the reaction between them to occur, they need to physically come into actual physical contact, right? It's just imagine yourself, like again, during the recess, running around this classroom and try and find another one. So it is pretty hard. But during the lecture, sitting next to each other, it's a perfect time to talk because because you're in the ideal orientation, right? So to actually next to each other, I mean, perfect time for interaction. So the same as with the reactions. So when the two molecules are in solution, for them it's hard to find each other. It's not only the energetic part, right? But it's also the relative orientation and distance between them. So when they bind to the active site of the enzyme, they become next to each other. So the enzyme already orients them. And that is what we call the entropic component. So the enzymes not only decrease the energy, I mean help decrease this activation energy, but also orient the substrate in ideal geometric orientation relative to each other so that the reaction can go. Again, this may seem a little disconnected from what I was talking, but this is just an example how the entropy can contribute to the rate of the reaction. Here, there is neither of these three reactions that I'm showing you is enzymatically catalyzed by the enzyme. This is just, I mean, a reaction going on in solution. So chemically, all the same reactions. So it's interaction between the ester and the carbonic acid. So if we take these two molecules and measure the reaction rate, whatever the number is, let's give it an arbitrary value of 1, right? Then, if we will have this molecule, which is physically the same as that one, but just imagine that these two guys were tethered together, right? So, but they still can, it's kind of this ethyl ester is tethered to this acetic acid, basically, right? But they still can, I mean, move relative to each other. So just by tethering them, or you can call it, I mean, putting them on a leash, you already accelerate the reaction by 100,000-fold. And this is purely, there is nothing to deal with the energy. So it's just the entropic component. So the entropy dropped so significantly that the reaction started to go 100,000 times faster. But then, if you decrease this rotation, actually, if you remove the rotational component, right, so that they will not even rotate relative to each other, so you will get 3 more orders of magnitude. So you will gain another 1,000-fold enhancement. So that's just an illustration of how important is that to have the substrate to be properly oriented for reaction to go. So that is related to this. Okay. So what are the other factors? I mean, in the okay. So far we talked about two major contributions by the enzymes, right? So one is the decrease of the activation energy, which is achieved through multiple weak interactions between the enzyme and the transition state. Second is the entropic component. So that would be important for the reactions. The entropic component is important mainly for the bimolecular reactions and up, right? So when this one or two substrate needs to be next to each other. Another important thing is the desolvation. So it's a displacement of water molecules that would otherwise impede the reaction. A good example, I'm not sure if we'll talk about this later, but if we go, here we go. Hexokinase. So that's an example of the desolvation. So during the reaction cycle, the glucose molecule and the ATP get closed in a little cavity inside the active site of the enzyme, right? So water is being excluded from there and being stripped off from the glucose and from the ATP, so that the reaction doesn't have any choices. I mean, so there is no. So the ATP will not be hydrolyzed by water molecule, but will be transferred onto the glucose. So by doing this, the enzyme basically, so what the enzyme is doing, it's basically desolvating the substrates. So it's just removing the water molecules from the substrate. But it essentially accelerates the reaction rate because they will have no other choice but reacting with each other. Okay. So in other words, when the substrate binds, many substrates are hydrophilic molecule, right? So they are surrounded by water. So, and when they bind to the active site of the enzyme, all these water molecules being stripped off. So they get back to solution. This in itself contributes to the energy, right? Because I mean, this is release of energy. And also it makes, it sort of liberates the chemical groups which otherwise would be shielded by the water. Yes, also it's important to actually have everything oriented in the active site, especially for the electron transfers, for example, so that there will be no distortions. Quite often enzymes cause so-called induced fit, so that so induced fit is when the active site of the enzyme is being actually changed while the substrate is binding. So the substrate shapes the binding pocket of the enzyme. And quite often the active site of the enzyme chemically participates in the reaction which is being catalyzed. So it provides group, as we will see at the end of today's lecture. It provides some groups. I mean, but then is being actually restored. Okay. What are the factors that affect the activity of the enzymes? One of the most important factors, as for any chemical reaction, would be temperature. So for the inorganic reactions, quite often the increase of the temperature, right, will have almost linear dependency, right? So the higher the temperature, the faster the reaction, and it can be linear. I mean, for pretty like for a good range of temperatures. So it's not the case with the enzymes. So with the enzymes, we always have this asymmetric bell-shaped curve. So any ideas why we have actually? Well, it's clear that on the left-hand side, I mean, the enzyme, the rate of the reaction, the speed of the reaction will depend on the temperature, just because, I mean, the higher the temperature, the faster this reaction and the faster the enzyme will do it. But why is it dropping abruptly here at the high temperature? Exactly. So because we talked in the beginning that the enzymes are proteins, and the proteins have tendency to be denatured at high temperatures. So then we'll have this abrupt drop in the activity of the enzyme, not because the reaction doesn't go, but because the enzyme is being killed. Or it would be more right to say that the active site of the enzyme no longer exists, right? So there are lots of examples of the enzymes, like RNase, for example, right? And enzymes which have the number of disulfide bonds, which even when it denatures, right, so it loses, I mean, its 3D shape. But then when you drop the temperature back to the room, so due to those crosslinks through disulfide bonds, it goes back to its native conformation and regains the activity. So for some enzymes, this would be irreversible, but for others, it can be reversible. Okay, so the same as the temperature would be pH. So obviously enzymes need their functional groups in particular protonation states, especially those enzymes which catalyze acid-base type of reactions. Okay, ionic strengths. I mean, here just, I mean, not only the particular metal ions, but just in general. So, and presence of cofactors and prosthetic groups. So, presence absence of coenzymes would have all or nothing effect. So without them, no reaction. With them, reaction goes. pH, temperature, ionic strength would have a bell-shaped curve, right? So that there is an optimum, but there is like you go low, you go high. I mean, that would be suboptimal. So most of the enzymes for a range of different conditions will have this bell-shaped curves. Okay, so what are the main types of the catalytic mechanisms that the enzymes can do? It's very simple. I mean, it's same as in organic chemistry. So we have acid-base reactions, right? So which is a transfer of protons. So we have covalent catalysis, and here we will talk mainly about the something like redox reactions. There is metal ion catalysis mechanism is when the metal takes active part in the catalysis of reaction by the enzyme. And there could be even electrostatic catalysis. I mean, so that is when the particular charge is important in the transition state. So you probably saw something like this already a number of times in the first module. Okay, so how do you think which of these amino acids would be participating in the general, could participate in the general acid-base catalysis? Huh? Which are the acidic ones? Exactly. Okay, these ones. Okay, so why acidic ones? Okay, yeah, that's good. Okay, so by definition, the logics, I mean, is, so in order to participate in acid-base catalysis, you need to be able either give protons or take protons, right? So now we talked about the ones which can give proton. So these are aspartate or glutamate. Okay, what about the ones which can take proton? Okay, which are those? Okay, yes, so they are right here. So these guys can actually accept a proton, and well, here they're already shown in a protonated form. So that's how they would look like. So any of these 5 amino acids would be participants in the general, like in the reactions involving transfer of the protons. Okay, why not, for example, these guys like serine or threonine? Have OH. Can it be protonated? No, that's right. So they actually will see that they do participate sometimes in the acid-base reactions, but not as active, let's say, but not as a key participants because they cannot be protonated. So water molecule can be protonated, but not OH group. It has pKa of something like 16, which means that is it 16? Yeah. So in other words, I mean, so no, at physiological pH, you cannot deliver the proton there. So, but if the proton comes on one side, immediately it sort of leaves on the other side of such group. So we'll see it now. Okay, this is a general acid-base catalysis. We already actually talked through it. All right. So how that happens? I mean, so in most cases, it's the proton transfer mediated by weak acids or bases other than water. So it actually, like for example, the aspartic acid in the active site of the enzyme at physiological pH is usually deprotonated, so it can take a proton from, for example, a water molecule, and then result in hydroxyl, becomes a very strong nucleophile, and being a strong nucleophile can basically do the nucleophilic attack onto this carbonyl carbon, right, and hydrolyze this peptide or whatever is shown here. So as a result, we'll have That's how the like peptidases work. Okay, covalent catalysis. So here we're talking mainly about the reactions such as the nucleophilic attacks. So in the nucleophilic attack reaction, there is usually a nucleophile and electrophile. So what are the most common nucleophiles? Actually, the most common nucleophiles would be the for example, a lone pair of electrons of the amino group, like this one, or the O minus radical, or the S minus, or the carbanion. So, but we will see that these are almost super versatile. So when you have lone pairs of electrons on the amino group. So the electrophiles are such as phosphate. So the ones which likes electrons, which have partially positive charge, right? So they like electrons coming in. So it would be, but the most common, of course, it's carbonyl carbons. So because they connected through double bond with oxygen, which has partially negative charge, right? So the electrons been pulled towards the oxygen, so that carbon has partial positive charge. As a result, it's lacking electrons. It really wants to get some electrons, and so any nucleophile, such as let's say amino group with this lone pair of electrons, would be a perfect attacker. So metal ion catalysis involve metal ions which are tightly bound in the enzyme complexes with the substrate. So there, quite often, this type of catalysis happens. It's when the electrons have been transferred, like in the electron transport, for example, or during the photosynthesis and many other reactions. Okay, I think actually I would like to skip to the end where we'll talk about the chymotrypsin as an example. So usually students get a little scared, about that part, about the mechanism of the chymotrypsin. I'll tell you up front, so I'm not expecting you to know the mechanism like step by step. That's not required. What's important is the conceptual types of the reactions which occur there, which we'll quickly go through. Well, first of all, the chymotrypsin it's a peptidase which is secreted basically in the intestine. So we have it. Almost all mammals have it. This peptidase basically digests the proteins from the food that we consume. So this enzyme. Here's just the secondary structure diagram of this enzyme. So it has a number of these disulfide bridges, which keep it in one in a particular three-dimensional shape, which is important for its activity. So it has the active site and the hydrophobic pocket where the substrate is binding. So it's known that the chymotrypsin actually cleaves the peptides or proteins after hydrophobic amino acids, such as phenylalanine, for example. So, and this hydrophobic pocket is needed for those hydrophobic amino acids in the peptide or a protein to be cleaved to enter and bind to it. Okay, how the things happen? So here's the active site of the enzyme. So here's the peptide that needs to be cleaved. So again, it has this hydrophobic phenylalanine. So it binds to the active site of the enzyme. So what happens next? So first of all, there is this important serine in the active site of the chymotrypsin, right? Which is a good nucleophile, right? So, however, it can become even a better nucleophile because of the presence of this histidine. So what histidine is doing? So it pulls that proton. It cannot take it, right? Because OH will never give it. So it pulls that proton. It kind of and holds it, and that actually makes this even more electronegative. As a result, this one would be a much better nucleophile than just the OH group at the serine. So okay, then this oxygen nucleophilically attacks onto this carbonyl carbon group of that phenylalanine residue in the polypeptide chain. The result of this is this so-called tetrahedral intermediate. So when we will have this peptide covalently attached to the serine, but this is a very short-lived transition state intermediate. So basically it collapses almost immediate because of redistribution of the electrons and collapse of that bond, mainly, like the bond between the carbonyl-carbon and the amino group in that peptide chain. So as a result, we will have release of the C-terminal portion of the protein that is being cleaved, right? So this one is now free, it can go. So in the second part of the reaction, now what happened is that we got the N-terminal portion of the peptide, which has got stuck in the active site of the enzyme, which is covalently bound to the, through that serine residue, right, to the active site of the enzyme. So what happens next? I mean, the water molecule comes in. The water molecule itself is a very bad nucleophile because, I mean, it has these 2 protons, but all what you need to do is just strip 1 proton away. And that's, again, that's a job for the histidine. So the histidine takes that 1 proton away from the water, and then we have a hydroxyl. So hydroxyl is a much stronger nucleophile, which now does the same as what the serine was doing in the first part of the reaction, right? So, and as a result, what we again have the tetrahedral intermediate, so, which is then, after redistribution of the electrons and collapse of that bond with the serine, right? So here, so this bond is being collapsed. So we will have the C-terminal portion of the, oh, I'm sorry, actually, it's because it goes here, so this is the result of that. So once this redistribution occurs, so that basically the N-terminal portion of the peptide is being released. So when we regenerate the active site of the enzyme. I know it sounds a little complicated, but again, I don't want you to know step by step. The key things here is which amino acids serve as the nucleophiles. Serine, right? So, and by itself, serine is not such a super strong nucleophile, but there is a histidine which always wants to pull that proton and sort of make it stronger nucleophile. Which amino acids are givers and takers of protons here? So these amino acids are like histidine, for example, right? So these are, so here, the chymotrypsin is an example of the enzyme which has all kinds of catalysis in just one enzyme. Here we have an acid base, we have covalent catalysis. Well, there is no metal actually in the active site. I mean, so from that perspective, it's not really like a metal ion type of catalysis, but there is also electrostatic portion here which is involved. Like for example here, this glycine, right? So it also helps to coordinate, I mean, this through electrostatic interaction with this oxygen, right? So it helps orient it properly. So as a result, I mean, this oxygen, it's called oxyanion. So it actually has been stabilized there in the transition state. So these are the interactions which the Linus Pauling was calling those multiple weak interactions in the transition state. So the interaction between the glycine, between the main chain amino group of serine, so like even the histidine. So these are all those weak interactions in the transition state. So from the chymotrypsin, really want you to remember the like what are the nucleophiles, what are the electrophiles, who is the base, who is the acid. So, okay, yeah, so. All right, so we can, let's see what are the main points that we went over. Well, we know that the enzymes are super important for life. So that they're not, the enzymes are proteins, right? But there are other types of catalytic molecules in the living organisms, such as ribozymes, which are catalytic RNA molecules. So that the many enzymes are not just polypeptide chains, they contain coenzymes, which will be either in the form of a cofactor, which comes and goes, or a prosthetic group, which always sits on the enzyme. So the enzymes have their classification, so it can be named either based on the reaction they catalyzed or just historically, which is called a trivial name. So the enzymes are they're very highly effective catalysts, and they accelerate the rates of reaction more than what the inorganic catalysts do. So the main contribution of the enzyme to the catalysis is through formation of this enzyme substrate complex, and then pushing it to the transition state. So because the active site of the enzyme it has the highest affinity, or in other words, most congruent to this imaginary transition state, I mean, a very short-lived state, I mean, in the path of the reaction. So the enzyme, by forming multiple weak interactions with that transition state, it actually pushes the reaction to that state, and as a result, I mean, the reaction goes faster. So the active site, yeah, we know what the active site is, and we know what the delta, the activation energy is. So, so that the higher the activation energy, the slower the reaction. So what the enzyme is doing is just lowering it through establishment of those multiple interactions. So that the key thing to remember that the equilibrium is not affected. So, so the equilibrium is the concentration of the substrates, basically. So, okay, so. So the significant part of the energy in the transitions, I mean, for formation of these multiple weak interactions in the transition state is hydrogen bonds, sometimes ionic interactions, electrostatic interactions, hydrophobic types of interactions. I mean, these are but when you have many of them, that's what makes a difference. So it's not that one particular bond makes a huge difference. It's only this one. Okay. So I guess that's it for today, and well, you can review the summary by yourself. Have any questions? I'm here.
Кристина.
Очень приятно.
Расскажите мне, я слышала, вы, я видела, вы закончили Ель.
Я не закончил, я там работал. Ой, сейчас я. Там работал.